Okay, let's start recording. First, we will do the quiz. Let's see. Okay, we have got a bunch of image here. So, I think this is a picture of a cell, if I am not wrong. Okay, which label structure packages and ship protein in vesicles to their destination? Okay, it's EES. I think this is the smooth endoplasmic reticulum. E. But what it does, I don't think I have any idea. Let's go with F. This part, that's the thing. Which labeled organelle is considered the powerhouse of the cell? I think that's the mitochondria K. That's the only thing I know. This labeled organelle is known for its ability to synthesize lipids. Okay. It's a bit strange. Let's see. This labeled organelle is known for its ability to synthesize lipids. Okay. No, maybe B. The arrow at C to an organelle that is home to genetic material and facilitates the majority of the cell's operation. What is the name of this organelle? Okay. At C. So I think this is the nucleus, right? So I think this is the answer. Which labeled organelle digests worn out cell parts? Okay. I have no idea. It's A maybe. Who knows? Okay. Region G is an interconnected network. What process mostly occurs? I know only one thing. G. I have no idea what is this, but I know transcription and translation is an important process, so maybe this. I don't know. We'll see. Which label organelle creates ribosomes? Okay. Which labeled organelle creates a ribosome? It's a no idea what is ribosome. We have to see. I have no idea. Let's go with B. The structure marked B organizes the spindle fibers during cell division. What is it? Okay. A. What is A? I think this is the centriole, if I am not wrong. We'll see. Okay, which combination correctly lists the three components of a single RNA? RNA consists of, I think, a phosphate group, maybe this, because I think DNA consists of a deoxyribose sugar and RNA consists of a ribose sugar. Which statement best distinguishes RNA and DNA? Okay. Which statement best distinguishes RNA from DNA? Okay. RNA contains base thymine, whereas DNA contains base adenine. Is it true? No, I think RNA contains. RNA contains. I don't think so. I think this one is true. RNA has one strand, whereas DNA has two strands. So this might be true. We will see. Which statement best matches each RNA together? I think cytosine and thymine. Is true, and another one must be uh, uh, I have no idea. Maybe this uracil and adenine, because AT and EU. Which of the following statement is true of codons? They are present only in DNA as RNA. Encoding can specify several. The cell type, a sequence of 3-letter genetic sequence found in both DNA and RNA. Each codon is made up of. I have no idea. Maybe this. We will see. Which of the following sequence correctly describe the flow of genetic information? When transcription happens, then translation happens. Yes. Which of the following correctly identifies the difference between transcription, translation, and prokaryotic cells? No idea. Maybe this transcription converts DNA into an RNA copy, whereas translation reads the. We will see. In plant cell, which of the following structure protects the cell and give it shape? So I think that's the cell wall. Which of the following processes only occur in plant cells? Or photosynthesis. Which of the following correctly identify the similarities between both? No, they are definitely not same cell. Both have cytoplasm. We will see. Which base is not present in RNA? But I think that's thymine. Which of the following correctly describe the sequence of 4 different types of protein? I think primary, secondary, tertiary, quaternary. What are the building blocks of RNA called? I think nucleotides. Okay, this is something different. So they have asked in the lab, your assignment is to determine the DNA template strand that encodes a protein with the amino acid sequence MET, HIS, GLU. Okay, what are these? I have no idea. So here's MET. So I think then if it is immediate, then there should be AUG, and then it said HIS. So what is the question? I have never got the question in first place. Let me read the question first. What are all the possible codons for the mRNA strand? Separate each codon with a space and each permutation with a comma. Okay. Okay. HIS, where is HIS? Yeah. Then CAU. So CAU, and then GLU. So GLU is this GA. This is one answer. And there could be AUG, and then CSC maybe, and GLU, GA, and so many are possible. Similarly, AUG, and then CSC, and GAA, G. I am writing this only 3. Okay, using the mRNA sequence you obtained, what would you expect the codons of one of the tRNA strand to be? Okay, I have no idea. So if it's A, no idea. Which of the following information aforementioned would one possibly originate DNA simple strand P? Separate each anticodon. No. Okay. After you finish, the teacher assigns another protein with the following mRNA codon sequence UUG, CCC, GAG. Translate each codon and write the correct amino acid sequence. Write your answer with a string of letter abbreviations of amino acids separated by a hyphen. Okay. UUG, it's LU now. I think it should be LU. And then CCC is for pro. And then it's given GAG. GAG is glue. If your mRNA codon sequence experiences a mutation change in the codon from GAG to GAA, okay. GAG to GAA, both belongs to the same glue. So what will this mutation have on the resulting polyprotein? I think there will be no change. It remains unchanged. So I think it's finished. Okay, let's see how we did. Not bad. 17 out of 25, and most of the things I don't even remember, so that's good. Okay, this one is correct. Okay, let's take a 5-minute break, and then we will start dissecting and learning from the model and see how the model helps us. Okay, we can start dissecting the results of the quiz. So let's see. Let me go to Google Gemini first. Paste the picture. You help me. Presenting the figure. Also help me understand their role and importance. Let's see. So the model is currently thinking. Let's go back to the quiz results. You can see the labeled organelle is known for its ability to synthesize lipids. Okay, so shows the answer is E. With this one. Okay. Which labeled organelle digests worn out cell parts? Okay, that's D. Okay. Okay, region G with the network with ribosome and with ribosome batch, which was okay. The protein synthesis. Region G. Okay. Okay, I have clicked protein. It should be photosynthesis. Made a silly mistake here. I can count that as a plus one. So I was right. My answers. It's not possible to write all the things. It's okay. I got the concept right. Okay. So we'll see about this. This is correct. Okay, it remains unchanged. I have written that. So this one is also correct. Okay, so I got most of the things correct. So that's a good thing. Let's see. The model has given the answer. A centrosome. Okay. The centrosome is an organelle that serves as the main microtubule organizing center. A role and importance, it plays a critical role during cell division by organizing microtubules into spindle fibers, okay, which are essential for separating chromosomes correctly between the two daughter cells. Okay. B is nucleolus. Okay. The nucleolus is a dense, prominent structure found inside the nucleus. Role and importance, its main function is to produce and assemble ribosomes. Oh, okay. The cell's protein making machinery. Okay. C is nucleus. Okay. Yeah, C was nucleus. The nucleus is a large organelle that acts as the control cell of the control center of the cell. Okay. Role and importance, contains the cell's genetic material, DNA, organized into a chromosome. The nucleus controls all cellular activities, including growth, metabolism, and reproduction, by regulating gene expression. Okay, yeah. Then comes the ribosome. So that's D. D is the ribosome. Okay. The ribosome are tiny molecular machines shown here as small dots, which can be found in the cytoplasm. Plasma reticulum. Okay. E is plasma membrane. E is no. I think E is. I think E is smooth endoplasmic reticulum. So H is the rough endoplasmic reticulum to them. No, G is the rough endoplasmic reticulum, and F is the Golgi apparatus as far as I remember. This in K is smooth endoplasmic reticulum. No, I think K is. This is definitely wrong. Is mitochondria. If I'm not wrong, let me see if it gets it right. That I am definitely sure of, that K is the mitochondria. Yeah. No, J is not. J is not. J points to the boundary layer, so I think J is the cell membrane. J must be the cell membrane. Okay, E, no. I think there is a mistake because this one is E, and it's definitely not the cell membrane. J is the cell membrane. I think he made a mess of it, but that's okay. No issues. And G is the Golgi apparatus. No, G is not the Golgi apparatus. F is the Golgi apparatus. I is the lysozyme sign. Okay, but I have my doubt. Is the ribosome. Okay, D is the ribosome. That's correct. I think most of the things are correct, and the rest of the things I know, because because I know E is smooth endoplasmic reticulum and F is the Golgi apparatus, and G is the rough endoplasmic reticulum. So I can proceed based on that. Let's see what we have done wrong. Let me ask. Labeled organelle is known for its ability to synthesize lipids. Centrosome or ribosome? Okay, smooth that would be smooth endoplasmic reticulum. Okay, so smooth endoplasmic reticulum is this E. The answer must be E. Yes. Let's see what we have done. Okay, let's ask, which labeled organelle digests worn out cell parts? So okay, that's the lysosome. So it is given the answer D. So lysosome is D. So these structures are the lysosome. Okay. Yeah, it's saying I, but I is not pointing towards this. I is not I pointing there, not here. So that's a mistake. Okay, got it. Let's see. Okay, region G is an interconnected network with a ribosome attached. What process are mostly occur here? Let's ask the model. Okay, it says it seems there must be a mix-up. So protein synthesis and protein folding and modification are the things. Saying the correct things. Okay, so the answer was D. Okay, got it. So the Golgi apparatus. Region G is so this is the rough endoplasmic reticulum, if I am not wrong. Ah, yes. Okay. So we were correct mostly. Next is okay. This was photosynthesis. I knew. Uh, this one is also I think I knew. So I'm not going here, but we should know. I should know a bit about this. So let me ask. Let's see. Let's see what the model gives, and then we will ask the question and understand all the relevant concepts required. Okay, so to determine the DNA template strand for the protein sequence met is clue, you need to work backward from the amino acid mRNA sequence and then to the DNA template. Okay. Can you can you give a basic overview and all the important concepts required to understand this process as okay, let's see. I think what it will give is will be very helpful for us. So let's move forward. Okay, so DNA, it's the deoxyribonucleic acid as the master blueprint for an entire organism. It's a long double helix molecule, contains a 4 alphabet A, G, C, T. Okay, and proteins are the worker of the cell. Proteins are genes and laborers of the cell. They do almost everything from finding structure like collagen in the skin to speeding up chemical reactions as enzymes. Proteins are okay. Smaller unbuilding blocks are called amino acids linked together in specific order, like beads on a string. The sequence of amino acids determines the protein's unique 3D shape and distribution. Here the flow of information from gene to protein. Transcription, gene to mRNA. Okay, in transcription, what happens? It goes from DNA to mRNA. This is the process of creating a portable copy of a gene. Okay, A segment of DNA is unused. An enzyme creates a messenger RNA molecule that is complementary to the DNA's template strand. Okay, understood. This mRNA is like a photocopy of the blueprint. It's a single stranded molecule and uses uracil instead of thymine. Where DNA has an A, the mRNA will have a U. Okay, so for A we will get an U. For G we will get a C. Okay, okay, understood. In case of DNA we should have got a T, but in case of RNA we will, since it's an RNA, we will get a U. Okay. Okay, next is translation from mRNA to protein. So it's basically DNA to mRNA, then mRNA to protein. This is the process of reading the mRNA copy to build the protein. The mRNA leaves the nucleus and goes to a cellular machine called a ribosome. Okay. The ribosome reads the mRNA instructions in three-letter words called codons. For example, AUG is the codon. Each codon corresponds to a specific amino acid or a stop signal, okay, which you can look up using a genetic code chart. Okay, I think there was something. Yeah, these are UA, UAG, UGA. These are the stop symbols. Okay, understood. Then each codon corresponds. Okay, the ribosome links the amino acid together and in the order specified by the codons create a protein. Okay, key concept for the problem: codon, a 3-base sequence on mRNA that codes for one amino acid. Okay, genetic code, the set of rules used by cells to translate codon into amino acids. Okay, the set of, okay, redundancy or degeneracy. The genetic code is redundant, meaning that most amino acids are coded by more than one codon. Okay, for example, in the chart, CAU and CAC both code for histidine, which is why a single protein sequence can come from several possible DNA sequences. Okay, understood. Let me ask what is actually is codon and anticodon, because I think it's required to answer these questions to have a sound knowledge of what is codon and what is anticodon. Okay, we have got a response. So in simple terms, codon and anticodon are sequences of 3 letters in the genetic code that work together to build proteins. Okay, they are essential for translating the genetic instructions from a gene into a functional protein. Oh, okay. Codon. A codon is a specific sequence of 3 consecutive nucleotides on a messenger RNA, mRNA molecule. Think of it as a 3-letter word in the genetic instructions function. Each codon specifies a particular amino acid that should be added to the protein chain. For example, the mRNA codon AUG codes for the amino acid methionine. Okay. Special codons, they are the start codon and the stop codons. Okay, like AUG, UA, UH. Okay. Okay, what are anticodons? An anticodon is a corresponding sequence of nucleotides found on the transfer RNA molecule. It is complementary to the mRNA codon. Okay. Okay, I think I understood. function. The job of an tRNA molecule is to act like a taxi, carrying the specific amino acid to the ribosome. Okay, the anticodon of the on the tRNA ensures that it parks at the correct mRNA codon, delivering the right amino acid at the right time. Okay, how they work together during protein synthesis. The mRNA molecule is fed through a ribosome. Each codon on the mRNA is read, a tRNA molecule with the matching complementary anticodon binds to it. For example, if the mRNA codon is AUG, the tRNA with the anticodon UAC will bind to it. Okay, the specific tRNA carries the amino acid methionine. Okay, which is then added to the growing protein chain. Okay, the precise pairing between codons and anticodons ensure that the amino acids are assembled in the exact sequence dictated by the original gene, resulting in a correctly formed protein. Okay, so let me ask, what are these? Will you? What are L U G L U etc. in the figure? I think I have a basic idea of these, what are these things are, but I think it's more important to understand. Okay, these are the building blocks of the protein. Okay, amino acids are small molecules that link together in long chain to form proteins. Okay, think of them as the individual beads. Okay, the genetic code chart you are looking at, it is essentially a dictionary that translates the language of mRNA, written in codons. Okay, into the language of protein. Each three-letter codon on the mRNA molecule directs the cell to add a specific amino acid to the growing protein chain. Okay, here are a few examples of the chart. LU is leucine, GLU is glutamic acid, PHA is phenylalanine, SER is serine, MET is methionine. Okay, okay, understood. The specific sequence of these amino acids determines the unique structure and function of the final protein. Okay. Let me see what other things we got wrong. Okay, this one is correct. I just can't write all the possible combinations. Using the mRNA sequence you obtain, what would you expect the codons of one of the tRNA strands to be? Separate each anticodon with a space. So let me ask this. But from the answer, I think it's clear. Uh, if it's AUG, so A will be U will be A, and G will be C. So, and CAU will be C will be G, A will be U, C will be A, so on, and GA will be G will be C, and A will be U. So, yeah, understood. I think it's the mRNA sequence where G will be replaced by C and A will be replaced by U, and vice versa. And since there are no thymine in mRNA, so in case of T there will be U. And let's see what, using one of the possible mRNA sequences from previous problems, the corresponding tRNA anticodons would be UAC, GAU, and CUU. Okay, determine the anticodon. The anticodons on the transfer RNA tRNA are complementary to the codons on the messenger RNA. They pair up during translation to ensure the correct amino acid is delivered. Okay, A pairs with U, okay, and G pairs with C. Understood? Here is the big one for this. Yeah, I have understood the concept for this. Okay, with all of the information aforementioned, what would one possible original DNA template strand be? So from the mRNA to DNA, we are going back. So in this case, I think U will be replaced by T, and G will be replaced by C, and vice versa. And A will be replaced by U will be replaced by T, and A will be A, I think. As it's the case, A will be T, and U will be A, and G will be C. Yeah, it's correct. So let's ask, just to be double sure that the model agrees with the concept I have learned or not, or I have understood the concept correctly or not. So yeah, so one possible original DNA strand would be TAC, GTA, CTT. So I think this is what we got. Yeah, TAC, GTA, CTT is correct. Deriving the DNA template. While your question mentions anticodons, the DNA and mRNA tRNA molecules. Here's how we find the DNA template strand from the mRNA sequence: AUG, CAU, GAA. mRNA sequence. This is the messenger RNA that codes for the protein AUG, CAU, and GAA. Okay. DNA template strand. This is the original DNA strand that the mRNA was copied from. It is complementary to the mRNA. Okay. The mRNA codon AUG comes from the DNA triplet TAC. Okay. Yeah, got it. And similar for CAU and similar for GA. So this gives the original DNA template TAC, GT, and CTT. For clarity, the corresponding tRNA anticodon QAC, GAU, and CUU, which we got earlier. Okay. I think it's a lot of things to take in, so you can revise one more time what are the things useful here. So this is the centrosome. I think this is the lysozyme, uh, which they, and this is the Golgi apparatus. This is the mitochondria. This is the cell membrane. This is nucleolus. This is nucleus, and this is the rough endoplasmic reticulum. This is the smooth endoplasmic reticulum. Yeah. And what is the ribosome? D. Okay. Here also we get what we've seen is transcription and then translation. From in transcription, what happens is it goes from DNA to mRNA, and then in translation goes from mRNA to protein using a tRNA. So that is it. I think one thing can ask about this also. I think this is the concept of degeneracy, which was the model gave earlier. So the mutation will have no effect on the resulting peptide. Understood. This type of change is known as silent mutation. Okay. Here's why it has no effect. Original codon GAG codes for the glutamic acid, and mutated codon GNA also code for this. Okay. So because both the original and the mutated codon specify the exact same amino acid, the sequence of the polypeptide chain remains unchanged. This is a common occurrence due to the redundancy of the genetic code, where multiple codons can code for the same amino acid. Yeah, understood. So I think we can take 10 minutes break, and then we can start with our next quiz and see how we do, whether the model helped us or not. So okay, see you in 10 minutes. So, I think we can start now with the quiz. Let's hope we do. Let's start. This is a completely new diagram. Yeah. But we can, if I whatever I can recall, I think this is the mitochondria, and this is the Golgi apparatus, and this is the smooth, and this is the rough endoplasmic reticulum, and I think these are the ribosomes, small, and this is the lysozyme. This is the centrosome or centriole, whatever. I don't properly remember the name. And this is the nucleolus. This is nucleus. So let's start. Okay, which labeled organelle stores the cell's DNA and control most of the cell activities? That's G, the nucleus. Okay, which of the following organelle modifies, sort and packages protein into vesicles for transport? Okay, this square. Okay. Which one could this be? I have no idea. I have absolutely no idea. Which one could this be? Maybe. Okay, don't know. The labeled organelle's primary function to synthesize protein. Okay, synthesize protein. I think this is the ribosome, as far as I can remember. So ribosome is C. The arrow at D points to the cell's outer boundary. Which option best described? D. This region E is an interconnected network with no ribosome attached. Which process primarily occurs here? Region E. E is the smooth endoplasmic reticulum. I think DNA. Replication. I think this is a protein, as far as I can remember. Maybe this. I don't know what this. These two I'm confused. Go for this. Let's see. Which labeled organelle is the nucleolus? Nucleolus is the middle one. I think F. What is the main function of the structured labeled H? H. Okay. H. This is the rough endoplasmic reticulum. Okay. What does the rough endoplasmic reticulum do? I think that this is not true. I think this. This is mitochondria. Which of the following structure helps organize microtubules during cell division? Microtubules during cell division. Okay. this centrosome? I have no idea. I'm doing A maybe. Okay, select the combination that correctly lists the three components of a single DNA nucleotide. Deoxyribose sugar. Which statement best distinguishes DNA from RNA? DNA is single, no. DNA contains base uracil, no. DNA contains ribose sugar, no. So this is because these three are false. Which statement best matches each DNA base together? Cytosine when think a codon is defined as, okay, I think this, or this, a codon. Okay, I think I'm a bit confused. Three nucleotides as far as I can remember. Which sequence correctly traces the flow of genetic information from DNA to the final protein products? So it's DNA to mRNA to protein messenger RNA. I think DNA to mRNA to protein. As far as I can remember, this is. Which of the following correctly identifies the difference between transcription and translation in eukaryotic cells? Maybe this, I have no idea. In plant cell, which organelle uses chloroplast? Which pair of structure is present in typical animal cell but absent in plant cell? Okay. Centriole, as far as I don't. Which of the following correctly identifies the difference between prokaryotes and eukaryotes? Maybe this, I don't know. Select the nucleotide base found in RNA but generally absent in DNA. Uracil. Which of the following correctly matches each shape of the four levels of protein structure? Yes. The building blocks of DNA are called nucleotides. Okay, so that's another similar one which we faced before. So let's see. It's MET, MET is this AUG, and then it's LYS is AAA, and then ASN is, where is ASN? ASN is here, AAU. Writing just one. Let's see. So So using one of the mRNA sequence you obtained in the previous question, what do you expect the codons of the tRNA to be? Okay, tRNA would be, I think for A we will get a U, and then a A, then a C, then a U, A for U, and U for A. So TNS strand would be, it should be T, A, and C, and A is TTT, and then for the last one it should be TTA. So UUC. Okay, so we have to name the protein, I guess, just like previous one. So UUC is PHE, and then GCU is LYS, I guess. No, GCU. GCU is ALA. And then AGA. AGA is ARG. Okay. I hope AGA to AGG. AGA to AGG same, so I think remains or no change, maybe. I don't know how, which one it will check. No change, I think. Submit. Let's view our score. See, we have done well. Improvement 21 out of 25. We have made 4 mistakes, so we can see. Let's see. So these are all correct. Wow. So okay, so some mistake. Which of the following? Circular. Okay, so prokaryotes have circular chromosome, whereas DNA of eukaryotes is linear. Okay, this I didn't know. So correct. So this one is correct. I can't name all. This one is correct. So this one is also correct. I can't name all. They expect me to name all, but that's okay. The concept is correct. This is correct. So I've written no change. So this one is also correct. So basically only one of them is incorrect. Most of them is correct. Fine. So there has been a improvement, as I can see. It should stop the recording now.